messieurs les secrétaires perpétuels honoraires, mesdames et messieurs les membres de l'Académie, mesdames et messieurs, it's a great pleasure today to open the symposium of the Royal Academy of Medicine of Belgium, dedicated to the scientific, medical, and academic activity of Professor Jean-Michel Foidart, who is our honorary permanent secretary that just finished his term, and a number of extraordinary and special scientists and physicians came from far away, as well as from Belgium, to honor the activity of Professor Jean-Michel Foidart. I will not be very long, but I cannot stop myself in mentioning that Jean-Michel Foidart is a very high-level scientist in biochemistry and cell biology, which are basic areas of research in medicine. He has been contributed to many discoveries, for example, laminin, which is a textbook notion, and other things linked to membrane metalloproteases. So he is an accomplished cell and molecular and biochemical researcher that is also a very high-level clinician, and he was able to take fundamental observations that are done in a biochemistry setting and transform them into concepts that are now practically used by patients. So usually we all pay this tribute to the bench to bed transition, but in this case it's real. It happened, and it happened for patients not only in our clinics, not only in a small clinical trial, but worldwide. So it has been a great honor for me to serve in the Bureau when Jean-Michel Foidart was permanent secretary. And with that, I congratulate him for his activity, and I pass it over to Professor Georges Casimir, our current permanent secretary.
Dear colleagues, ladies and gentlemen, Mr. Honorary Permanent Secretary, dear Professor Foidart, allow me the familiarity of the first name, dear Jean-Michel. For 5 years, you have been the 14th perpetual secretary of this illustrious company since 1841. But times have changed. The number of annual advices of the company asked by the government has been increased by 4 in recent years. The pandemic has placed on the shoulder of the Academy major responsibilities that it has had to face quickly. The French community of Belgium, confronted with the planning of the medical offer, has entrusted our company with it. All these responsibilities and many more, such as maintaining the scientific level of our session, you have taken up with brilliance, intelligence, and fineness as it should be. It seems essential to us to thank you for having thus maintained, such as the Orders of Lords in England, the Sword of England at arm's length during the speech from the throne, the quality of the Royal Academy of Medicine of Belgium. And for this, of all solemnly give you the Medal of the Academy bearing the face of its founder, King Leopold I, and on the reverse your name as a badge of thanks from the company for rendered services. And the President will give you now the Medal of the Academy. We saw that honoring you went through the organization of a symposium on the scientific work that has fascinated you throughout your life and also resulted in the creation of one of the fastest growing Belgian pharmaceutical companies today. Mitra notably produces synthetic steroids, but not only. You will have always been, with much happiness and efficiency, a researcher, a teacher, a head of department, a perpetual secretary, officiating in all these clauses with ease and rigor. Your career has been a tremendous success, and the Academy can take pride in you for having been led by a remarkable organizer. But it seems to me judicious to let you now introduce this session with all the skills that include yours.
Monsieur le Président, Monsieur le Secrétaire perpétuel, Monsieur le Premier Vice-président, Mesdames et Messieurs les académiciens, dear ladies and gentlemen, I must say that I am extremely happy and honored to have so many nice people and friends today in this audience. I would want to really thank the Academy of Medicine for having organized this session, but you know, I am not very important. What is important is molecular biology, clinical sciences, and the future of our teaching and developing new activities in the field of human health. I would want to very briefly summarize the fact that my scientific career was really influenced by two very important scientists. One was George Martin on the right-hand side from the National Institute of Dental Research. I spent 4 years doing dental research. And on the left-hand side is Stephen Katz. Steve Katz was at that time a very young chief of dermatology at the NIH in Bethesda, Maryland, and both of them were really supervising my research. This research started really with the Danish scientist called Engelbert Holm. He captured these wild mice who had a small tumors, and he described that this tumor was really composed of a sarcoma. George Martin had really the impression that in order to identify new molecules and new important proteins, you have not to look at normal tissues, but at malignant tissues. He said, okay, when you have a tumor, you have a clonal expansion of malignant cells who produce always the same proteins, and they're all alike, and therefore you have a much higher chance to isolate new proteins. Let's make an example. In immunology, there were at that time in the 1970s many scientists who had tried to isolate the immunoglobulin and to understand what it was about. And no one had succeeded from immunoglobulin obtained from the plasma. It's only those people who were looking at myeloma or Waldenström's disease with the monoclonal expansion on one type of IgM or IgG who were successful to isolate and understand what is an antibody. So we started looking at the proteins contained in that little mouse, and it turned that I was really the first to develop antibodies to that and to realize that it was really locating the proteins from that tumor in the basement membrane. Basement membrane were unknown, and the composition of the basement membrane were unknown at that time. And I had the fortune to be associated with the discovery of laminin, and the very first paper on laminin was cited more than 3,000 times. And when I returned to Belgium, I started doing research on extracellular matrix and membrane metalloproteases. And my very first PhD student, who is now a member of the Academy of Medicine, I just called her today, is Agnès Noël. She deserves really to be nominated. I called her just a few minutes ago my first professional wife. She is really an outstanding scientist. She was very supportive of all the studies we were developing together. I was first her leader, and then she became my leader. She is really outstanding, and in addition, she is a very nice and sweet person. Then subsequently, many other members of the Academy nowadays visited the lab, performed their PhD studies or their post-PhD training. like Didier Cataldo, Vincent Castronovo, Rakik, who is an ophthalmologist. He is a basic scientist and he is a pneumologist. So we had the opportunity with Agnès to develop really basic sciences in the Faculty of Medicine at the University of Liège. But in the field of obstetrics, and I was teaching cell biology and molecular biology for more than 15 years, something like 17 years. Then I was offered to go back to the clinics and to develop a large department of obstetrics and gynecology. And again, there I benefited of the support and the help of a wonderful endocrinologist called Ulysse Gaspard, who is in the audience today. And my second professional wife, who is a clinician, is also a member of the academy. It's Professeur Nizol. Michel Nizol is a fantastic scientist, worldwide known for her expertise in the field of endometriosis. And all the other people, the little guy that you see at the bottom will speak here at the Academy, Krivelka, in just next Saturday. And of course, there are many other people who are kind of cited, but who were really members of the research team. And in the field of Estetrol, there are many people who are now working either together or in Mitra, like Maud Jost on the left-hand side, Céline Gerard, you've seen also Anne Gallet, Christelle Pecqueux. The three first are now members of the Mitra team. The Belgian people know very well Blancpain. Cédric Blancpain is really at the cutting edge of science in painting mice, and by using very sophisticated method, he can paint in vivo the luminal cells of the breast, the basal cells of the breast, normal or malignant, and any cell type that you want, he can specifically paint them with at least 12 different colors. So we are actively working, and we have dedicated €1 million to combine research, coordinated research with Christelle Pecqueux, Agnès Noël, Cédric Blancpain, and his co-workers, Alexandra Van Kemel. I must also cite a collaborative research that we are doing with another member of the Academy who is also in the audience, Philippe Lefebvre. Estetrol is a neuroprotective agent, it's a neurotrophic agent. We have shown that in just young mice in postpartum, and just after a few days after birth, those mice, when they are treated with Estetrol, are protected against brain damage upon hypoxia. When you place the mice in a hypoxic chamber, those little pups will die, and they have huge brain damage. If you give them Estetrol, it doesn't do anything. And they go well, and they feed, and they can find their mother, their way, they are intelligent, and so on. It forced us to say maybe this Estetrol could be a neurotrophic agent also for people who become progressively deaf. And Philippe is the chairman of Ear, Nose and Throat at the University of Liège, and we are actively working on Estetrol for the people who are losing progressively their capacity to heal. So, but today we are here to know and develop the session on Estetrol. Estetrol was really developed in conjunction with Mitra. At the time I created Mitra in 1999 with François Forniery. It was not the Belgian habit to do it. When I went to the director of the university to explain that I wanted to create a spin-off of the university, he said, I'm not in favor of that. You should stay away from the business people. They are not good. We don't want to have such dialogue. Says, just stay away from that. I decided to do it anyway. We just rented a small apartment in that building. It was very small. We had just one bedroom who was used by the secretary. And we first did develop an intrauterine system that would release levonorgestrel. I had €1 million, but you need to get it up to the clinic, you need 150 million. And we had no money, no cash to do it. So we were stuck. I didn't know what to do with it. And suddenly, Warren Buffett, who had decided to create a charity foundation called Medicine 360, said, okay, this system is really good. It's the best. I'm going to provide you the finances. I will open all US academies, all US university and research centers, and we will do it together with the benefits from the US women and from the US market. We will collect a lot of money that will be used to distribute freely this intrauterine system to the developing countries and to the poor women in the United States. So it was a fantastic part of my professional life, because when you go to FDA and you say, I work for the poor people, they say, well, you work for the Medicine 360 and Warren Buffett, that's great. But the greatest aspect was that I did discover this guy. He is in the audience, and really he has become a great friend. Professor Mitchell Kreinen, thank you for being with us today. That was the start of a wonderful friendships and collaboration. And it started with the intrauterine system, but subsequently to the development of the contraceptive and of Estetrol. And as you know, Estetrol is produced by the human fetal liver. It's human specific. It's not produced by other mammalians. Throughout evolution, E4 was absent. And suddenly, when the human separated from the other primates, E4 did develop by the development of specific enzymes that are present in the fetal liver and that will become silent after birth. It crosses the placenta. Okay, Jean-François Arnauld will tell you that, in fact, you find a little bit of Estetrol in the large monkeys, but it's only present 1% during the last 6 weeks of pregnancy. When in the human, it's produced, the human fetus produces about 100% more, and it starts from the 9 weeks gestation. The fetal plasma are about 12 to 24 higher. It has a very common half-life, and now, of course, we produce it from plants by synthetic pathways. In fact, the estrogens that we are developing, it's really a highly demanded molecule, because the currently used estrogens are very old molecules. In 1929, 1930, 1936, and 1941 and 43, where at the time you see that within a span of 15 years, all the estrogens were developed and used in the clinic, and there have been no significant improvement for almost 80 years. What makes Estetrol really unique? We are supposed to see Arnauld pictures, but Jean-François is there, so you can see not only the pictures, but the reality. Jean-François Arnauld is a fantastic cell physiologist, a molecular biologist. He works at the University of Toulouse. He has an INSERM unit. We became friends. We met by accident in 2009. We became very much friends, and since we are working on Estetrol, and he was really the person who did decipher the mode of action. And as you know, Estetrol is really activating the nuclear estrogen receptor alpha, but is blocking the estrogen receptor alpha at the cell surface. He will explain that to you. And finally, we did develop this since 2015. I have retired from the university. I'm closely interacting with the Mitra people. I'm still a member of the board. And there are many, many other people who contributed to work. I must say that I'm also very happy of my last professional wife, who was my last PhD student. I had about 98 PhDs during my career, and Dr. Moonen, or it's there, Marie Moonen, is there with us, and I'm very pleased to say that almost 2 more and I would have had 100 PhDs. But you are the last. Thank you for coming. And finally, I want to say that nothing is really possible if you don't have also a personal life and a happiness in you with your wife. It's my best journey, and I used to say it's my best action in my life is to have been selected by my wife. And those are my four children who were very supportive. Thank you very much to all of you for being today and for celebrating the journey to Estetrol. Thank you.
Lecture, and I want to say that all the students that may watch us, the key paper on laminin was published in Journal of Biological Chemistry, right? And it is one of the highest cited papers in cell biology. There are examples of Nobel Prize winners that published in JDC, like Rao and Goldstein. So I think that if the science is really, really good, that's what's important, not running after impact factors and keeping the data for years in the drawers. And I think that's really, really important. Now, we have a wonderful session ahead of us, an exciting session with a lot of science.